Hi everyone, welcome to another episode of my Galaxy tutorial for bioinformatics. In this video, we are going to look at how to develop a Galaxy workflow for genome assembly. All right, so for this tutorial, the only requirement is to have an internet browser, and of course, you need an internet connection. So I'll show you everything step by step. All right, so this is an overview of the workflow we are going to build on Galaxy. So we will get our FASTQ files. I'll show you where to get them. We are going to process them. We will run QC on the raw reads, and then we are going to generate reports. All right. Now with the raw reads, we are also going to trim them as part of the QC. And then after trimming, we will get our FASTQ files. These FASTQ files we have, these are trimmed. We will run genome assembly on them, and then we will generate our context. So here we are going to use the tool Spades, by the way. I'll show you the tools, and for trimming we will use FastP. So for the trimmed reads, we will run QC on them again. That is FastQC, and then after that we will generate our reports. Okay, so here we will run two tools, FastQC and then MultiQC. So I'll show you all the nitty-gritties as we proceed with the exercise. So in Galaxy, this is how it's going to appear. We have it like this. All right, so that's how it's going to appear when we do it in Galaxy. So, but don't worry, don't get confused with all these things. I'm going to explain them to you step by step. So these are the tools that we are going to use in order to build the workflow. So we have the input dataset collection, flatten, FastQC, all this. I'm going to explain everything to you, but just know this overview. All right, that's what you need to do. So for the workflow pictures, I think I will share the link so that you can also have this in case you want to use them yourself. So this picture, I will leave it to you in the description box, the link to get this picture. Okay, so if you were to go to Galaxy itself, this is what you are going to see. Let's just check it here. So this is how we are going to build the workflow in Galaxy. So we are going to basically do something like this. So we will start from the input dataset collection. We are going to build everything gradually. Let's just go up here. We are going to build everything gradually, and I will explain these things to you, okay, so that based on the knowledge you get, you can also use that to build your own custom workflows or pipelines. Now, the reason why workflows are important is that workflows allow you to automate your analysis steps. It allows you to link the various steps. It allows you to automate the pipeline. And also, if you are handling multiple data sets or multiple samples or multiple files, all the more reason why you need to have a workflow so that you can reuse them. The workflow that we build, you can always reuse them. That means that if you have different batches of data sets, you can always run the workflow and then just use them. So that is what you need to know. Okay, so what we will do is to shift to the Galaxy interface and then proceed with the exercise. All right, so I am going to use galaxy.org. So for those who don't have the link, I'll leave the link to the Galaxy platform in the description box. All right, so there are different Galaxy platforms you can use. Use galaxy.org, use galaxy.eu, Australia, etc. So there are different servers across the globe, but I usually use the .org and the .eu. If you have any server, that's also great. All right, so this is what we have initially. Okay, so we are going to build the workflow. Okay, so let us do this. First of all, it's important that we have our example data set that we are going to use. And so I have already prepared some data sets that we will need. So let's first I repeat, I have already prepared some data set that we are going to use. So let's first import this data set and then we can proceed. So let's get the data set. I have it on one of my GitHub repos. So let's go to that repo, which is here. All right, so this is the page. I will leave the link to this page in the description box. I repeat, the link to this repo is in the description box. So we are going to assemble staff Oreos isolate. All right, so I'll also leave the link to the original paper in the description box in case you want to check them out. So when you click this, you have access to these links. All right, but if you want to take a look at the data sets, you can go to the ENA browser, just search the sample IDs. You can also go to SRA. All right, so for now we have the links. That's what we are going to use. So let's first copy this. When you are here, okay, when you visit this page, just copy the links, and then let's move back to Galaxy, and then let's come to the left side. There is the upload button here, so click it, okay, and then you can upload. And before we even click it, it's always important that you have a history and then you name it. It's a project. All right, so you need to always make sure you have a name for that project. So here, let's name this. Let's go to history, click plus. You get when unnamed history. So edit this one, edit it. When you edit, you can just say genome assembly, or maybe bacterial genomics or something of that sort. So you can just try any of these. All right, so once you do this, you get the appropriate name for it, you save it, and then once you save it, you can proceed. So now we need to get the data set. All right, so I'll just quickly go over again. So go to my GitHub repo. There's the page. I'll leave the link to this page in the description box. So you'll find this file here, staff Orioles patch 1.tsc. So click it and you get these links. So just highlight all of them, right click and copy, and then go to Galaxy, and on Galaxy go to the left side. All right, the left side here where we have tools, workflows, there's one button called upload, which is even here. Okay, so just search on the left side, you see that here. So click on upload. When you click on upload, you will see this window popping up. What you need to do is to come to fetch/data, this side here, this section. So click on paste/fetch data. All right, so when you click it, you are going to see this coming up. Okay, so when you click it, you should see something like this coming up. There's it. All right, so when you see this, just paste your links there. All right, we are using the paste/fetch data, so just click and paste your links in that entry box. You should have 6 links, and then click on start. So when you click on start, make sure the progress has gone to 100%, and you can check the right side, and you should see the files showing. All right, so you can now close this window and then check this. So you should see 6 files. All right, so we have to wait for the download to get completed, or let's say to get the data in our Galaxy accounts first, and then we can proceed. That's what we need to do first. So we should make sure we wait until the background turns green, and then we can now do the further processing and build our workflow. All right, so the files have been imported successfully, so we have them here. Okay, so what we need to do next is to convert the files to a collection, a data set collection. So what I want you to do is to just come to this side here where you have this check, okay, this box here. All right, so just click it. So when you click it, this, you should see these check boxes. Okay, they are going to be empty. All right, so what I want you to do is to check them. You can also use the select all here. All right, so just here you see select all. All right, so click on select all. It's going to highlight all of them. You can also just manually click each of them. That's fine. It's up to you. So make sure to highlight all of them, and then you will see this button here. It says all 6 selected. When you see this, there's a small arrow just beside it on the right side. Click it. When you click it, scroll down, and you are going to see this. Okay, select the build list of dataset pairs. This is what you should select. Build list of dataset pairs. I repeat, select build list of dataset pairs. So click it. So when you click it, you are going to see something like this. All right. So what you have seen is fine. You can give it a name for that collection. So you can just say FASTQ. All right. So you are going to get something like this. So let's name it as FASTQ. Or you can give it any name of your choice, raw read, whichever. But I think FASTQ is kind of simple and easy to remember and understand. So once you have the appropriate name, click on create collection. Create collection. So click it. When you click it, on the right side, you see that the 6 files are all going to be hidden. Okay, they are all going to be in this collection. When you click this FASTQ, you are going to see what? 3 pairs. Each of these, you will see the forward and reverse. All right, so this is what we want. Now we can go back, click on this FASTQ to go back. Let's go back. So we can go back to history genome assembly, click it, and it should be sent to this, and you should see just the FASTQ, this name here. And in it, we are going to have the description is even here, a list with 3 FASTQ sanga.gz pairs. This is what we want. All right, ladies and gentlemen, we now have our input data set, so we can now build the workflow. So to build the workflow, first, you need to have an idea of what you are going to do. Like how I show you the designs, I know what I want to do. So then it becomes easier for me to build the workflow. All right, so always make sure you have the list of steps, your algorithm, and then you now implement it with the workflow. So let's go to the workflow session. So on this page, come to workflow, this one here. So click on workflow. So when you click on workflow, all right, if this is the first time you are using that functionality, then there's going to be a blank space here. For me, there are things here because I've already built some workflows. All right, it's fine. These things do not matter. What we need to do is to create a new workflow. All right, so on this interface here, check this side where my cursor is hovering about. You'll see a button called create. Okay, create new workflow. So click on it. So when you click on it, you are going to see something like this coming up. All right, so now you see unnamed workflow. What I want us to do is to give this workflow a name. So come to name, and then let's say genome assembly. You can add any name, that's all. We can say genome assembly workflow, whichever, that's fine. Okay, so we are saying genome assembly, that's the name of the workflow. All right. So once you are done, so I'll repeat again. Once you are done, then come to this one here, this button here, this save button here, and then click on create a new workflow. So just click it. So once you click it, it's going to tell you that created new workflow genome assembly with 0 steps. So you should see this notification. It says success. So that is what we have. It automatically gives you a version as well, which is great. All right, so you can choose to keep it. That's fine. Later on, we can add something else. So we can also add annotation, which is a description of what this workflow is doing. It's optional. I would say workflow for assembly of bacteria genomes. Okay, so I can put it this way. All right, that's fine. And then whatever change you make, you can come back here and then say save workflow, and it will save the changes for you. All right, okay, good. Now time for action. We are now going to build our workflow. So we are going to start with this. We need to first start with inputs. It's a workflow, so obviously we need the input data. And so on the left side here, all right, you will see inputs. If you don't see it, just type inputs. and then you see it's here, this one here, input. That's what we need. So click it, and when you click it, you are going to find these options. All right, what I want you to do is to click on input datasets collection, the first option here. This is what we need. So input dataset collection. So click it, and when you click it, you are going to see what? Inputs. All right, so this is what we have. So I want you to drag it to the left because we have other things coming. All right, we have it like this. And then let's first give it a label. So on the right side, let's give it a label. All right, so I'll just say FASTQ. You can also say FASTQ. I mean the name, whatever name you want, just put it there. All right, so FASTQ. And I can even say FASTQ paired. Okay, so we can just give it this description to let us know we are going to input paired and width. Or you can just say FASTQ, and then in the annotation you can just say paired. This also works. All right, so let's keep it simple like this, and then scroll down to collection type. This one here, it's also very important. We need to define what we are going to input, the format. So click it, and when you click it, you are going to see these options coming up. List of data sets. We also have list of dataset pairs. We are going to use list of dataset pairs. This one here, list of dataset pairs. So click it, and when you click it, you should see list :paired. All right, I'll try to zoom in a bit. I think it's fine. I will just, let's see if I can enlarge it a bit. Okay, fine. This is great. Okay, good. So we see it here. So list :paired. Okay, now we are ready to roll. So we have our inputs. So I made mention that we will run FastQC on the raw read. So this input here is going to take the raw reads. And so we are going to first run FastQC. Now what we have here is a dataset collection. All right, so if you are going to run FastQC on it, you need to make sure you flatten it. So that means you have all the files bundled together. And so first we need to get this tool called flatten. Okay, so flatten, when you type flatten here, it will show here, flatten collection. So click it, this one here, flatten collection. So click it, when you click it, it comes here, just send it somewhere up. We are trying to also arrange our stuffs. You see it here. And then comes to a label, just type flatten. All right, so this will flatten it for us. Okay, and then annotation, you can put anything there. Yeah, maybe, yeah. So here I'll just leave it as it is. All right. Okay, so once you have it, don't forget to save whatever change you make. Try to be saving, okay, so that you don't lose some information going forward. If you do too many changes without saving, you tend to forget. So just a little activity and then you save. Now when you come here, let's drag it a bit here. When you come here, see this, where my cursor is, this circular shape here with this greater than symbol. I want you to click it, click in the middle, and I want you to drag. So when you drag, you will see this line. So drag and send it to the same structure or the same symbol in the flatten tool, which is here. So send it in there and then release. So when you release, this is what you are going to get. Okay, wonderful. We have this. So that means this is our input. So when we have the input, the data, okay, will be sent to the flatten tool. Okay, which is very good. Great. Now in the flatten tool, we are also going to do this. We are going to transmit this information to FastQC. All right. So what we need to do is to search for FastQC again, and then click on it. You see a FastQC read quality report. So click and you see it here. So drag it again somewhere here. You can also zoom in if you want. First, let's give a label. So fast QC. And then here annotation, you can give it a description, that's fine. All right. And then the same side here. All right. You can just scroll down just to make sure you have what you need. Here, we are not going to do any change here. Okay, just the name, the label, that's fine. All right. Now, when we come to flatten, you can just send it back a bit. And then this output, the output here. Okay. Before I proceed, let me say that for the tools, we have inputs and outputs. It's important. This one is already an input. That's why we have just an output. For the other tools, other utilities, we have inputs, outputs. So here, the flatten, we are taking its outputs, which is still going to be FASTQ files, and then we will send them to FastQC. This section, the raw read data from current history, this one here. We will send it there. This is what we are going to do with FastQC. We are assessing the quality. So this is what we have. So you also notice we have several lines, okay, branching from this or emanating from this. Is because flatten. So this tool, so the assumption is that you are going to have multiple files. So this is also an indication that you are going to have multiple files. Okay, so it's just descriptive. Now, when you are here with FastQC, you have your reports, a report per file. So if you have several files, to make it easier for you to read and compare, we can merge everything together. We will use the tool MultiQC. So on the left side, tools, clear and search for MultiQC. I've covered some tutorials on it, by the way, so just check them out. The links to those videos are in my description box. So MultiQC, it's here. Click it. It's also going to show. Bring it to the right side. You can just, if you want to move the workflow, just click on any empty space and shift, and then just move your cursor. So now we are here. So with MultiQC again, label, let's add a label. And then here, when we get to MultiQC, I want us to scroll down to this section, which tool was used to generate the logs. We need to set this. It's very, very important because MultiQC is used to aggregate files from different bioinformatics tools and analysis. So we need to specify which tool we are supplying its outputs to, MultiQC. So here we are using FastQC. So when you come here, you may see BAM tools. So click it and scroll down and search for FastQC, which is here, FastQC. So click FastQC to make sure we have that there. All right, so we should have something like this. Okay, so once you are done, I want you to come to the FastQC tool itself. There are two outputs, one for HTML, one for text file. So let's use the output for the text file, this one here. So what I want you to do is to drag the text file, drag it to MultiQC. This is what we need to have. All right, we need to have something like this. All right, for the MultiQC. Okay, so here we've done the QC for the raw reads. So we have the inputs, which should be in the form of data set collection. Okay, list of pairs. Then we flatten it, we run FastQC, and then we run MultiQC. So this is what we should have. Okay, good. Now this is for the raw reads. Now for the raw reads, we are also going to perform trimming. So here the assumption is that when you do trimming, it's going to improve the reads. Okay, of course, later on you can do your own stuff, but this is what I need to do. And also it's important that before you even do the workflow, you should do some prospecting. That means if you have multiple files or multiple samples, you can just take one or two, run FastQC separately just to see what data it is. And then based on that information, you can now use that to do a batch processing. All right. So here I'm going to trim all the samples. So when I come here, I'm going to use FastP for trimming. So when I come here, okay, MultiQC, I'll just type FastP. and it's going to come. So I'll click it and it's also going to come here. Note that there are several trimming tools available. So sometimes it can boil down to preference, or sometimes it can also boil down to the sequencing platform or some other criteria. So always read about them and see which one is appropriate for you. All right. So there's FASTP. FASTP will take in paired end reads. All right. So if it's going to take paired end reads, the output here, I can still drag it again and send it here. Okay. So there's it. Again, with the FASTP, click it and then let's add a label. I can put it like this, whichever name. At this point, the name, you decide what name you want. Okay, so this is FASTP. You can also say FASTP. That's fine. Any name is fine. Okay, just put in a descriptive name. And then annotation, that's information. You can put it there. Just some short notes. So let's say for trimming. All right. Now, when you come here, scroll down, the two parameters, it says what? Single length. We are dealing with paired end reads. So click and select paired end. Here we are using collection, so we will use paired collection. So click on paired collection. All right. So click on paired collection. It's very, very important. All right. So when you click on it, you'll notice something here. I want you to just undo it, okay, so that we can redo it again. All right. So what I want us to do is this. So I'll go over again. Once you select paired collection, you will see this red, okay, color coming here. It's a warning to tell you that the original information that we had, there's something wrong. So we need to change it. And so if we need to change it, then what you need to do is to make sure you come back to that link, that section here, this one here, and it even tells you to remove it. So just click to remove it, okay, because now the configuration has changed. We are using paired end reads. And then come back again and then drag it to the paired collections. Okay, drag it there. You should take note that if everything is okay, the lines are blue. If there's something unusual, it turns a different color, as we just saw. So now we have this here. So this is what you should have. All right. So take note of that. All right. So make sure you have this. Okay. Your labels, make sure you have your label. Make sure it's paired collection. Make sure that the line, okay, the output line connects to this text. Select paired collection. I will zoom in a bit for you to see. Make sure it's this way. Okay, you should see it this way. All right, good. Now that we have FASTP, what FASTP will do is to trim our reads. So when it trims it, we will have outputs. All right, we have output. So the output that we are interested in this section is the trimmed reads. Okay, we need that information. All right. So what we have to do is that with the trimmed reads, which are here, we need to run FastQC and we also need to run MultiQC. All right. So what we have to do is to select FastQC and MultiQC again. You can always duplicate it, but I want us to do it again so that we can get used to it. So let's come back to this section and then do it. Of course, what is also coming is a collection. So that means we need to what? Flatten it before we run FastQC, before we run MultiQC. So this is what we have to do. All right. So again, you can duplicate all these tools and then run them here, but I want us to redo it again. Okay, do it again so that we get used to. So I'll come back here to the tool, search for flatten, select it, and then bring it here, and I will give it a label, flatten. All right, here it's just the label that I want. And then when I come here, I'll put the output paired here. That's a flatten. All right, it should be something like this. All right, I can just send it here as well. So we should see something like this. Then I'll search for FastQC. Again, I'll make sure I leave a screenshot to this workflow in the description box so that you can always look at it and then just run it. All right. FastQC, I'll send it here. I'll give it a label. All right. And then I will send the flatten to it. I'll go to the left side. I'll search for multi-QC. I'll select it. There's multi-QC. I'll bring it here. When I bring it, I'll give it a label. All right. And then again, I'll scroll down. Which tool was used to generate? I'll click and then I will say what? FastQC. And then I will drag the text file outputs of FastQC into it. So we should have something like this. Okay. So this is so cool. Again, the screenshot for this workflow is in the description. So you can just take that and then use that to guide you. So we should have something like this. So we are done with the QC. Okay. Now that we are done with the QC, it's time for the meat. That is the genome assembly itself. So with the genome assembly, there are several tools available. Spades is there. We have Velvet. There are several tools. Depending on the sequencing platform you have, you can also decide to be specific. For example, Fly is used for long reads. We also have Unicycler for hybrid. If you have both long reads and short reads. I've covered several tutorials on genome assembly basically. So if you want to learn about genome assembly tools, just check the description where there's a playlist for that as well. All right. So now what we need to do is to now do the genome assembly. So we will come to the tools again and search for Spades. So this is the tool we are going to use. So Spades, genome assembler for genomes of regular and single cell projects. So I'll click Spades and then it should come. Spades, I'll put it somewhere here. It's fine. Okay. Now Spades, I'll come to label. I'll give it a name. Spades for, let's say, genome assembly. Description. And then scroll down. So when you scroll down, the parameter we are going to change is the single end or paired end short read. So I'll click and select paired end list of dataset pairs. This is what we need. All right, so paired end list of dataset pairs. So click it, and when you click it, you can come back to the FastP, which is here. And then the FastP outputs, let's put this here. The FastP output here, I want you to pull it to FastQ fast collection. Pull it here. And then that is it. All right, so we have our workflow, which has been done nicely for us. So now what we can do is to save. So we will save the now, as you are trying to save, it's likely you are going to get this coming up. It says saving workflow field, error duplicated step label flattening request. Don't worry, we are going to resolve it. This happens, all right? So I just thought I'll also bring this to your attention. So if you see this, it's talking about duplicates, all right? So the reason why we see this, let's click OK, and I will explain. The reason why we see this is that we have tools with the same name. Okay, so each of these, instead of tools, Galaxy classifies them as steps. Okay, each of these is a step. So we should have unique names for each step. So the reason why we are seeing the duplicates is that when we look at flatten, we give the same name. Okay, the same name flatten, the same name flatten. The same for FastQC, multi-QC. FastQC, multi-QC, because these are what? Repeated. And so what we have to do is to rename. Okay, so what we can do is that for these ones, flatten, FastQC, multi-QC, the first one, those for the raw reads, we will maintain the names, and those for the trimmed reads, we will rename them. So let's go to this one, I'm assist flatten. So click it and then add a value to it. We can say flatten 2. All right, and then save it. When you save it, you will still have the same error coming up. Okay, click OK, and that means we have to rename all the duplicates. So when you go to FastQC2, give it FastQC2. When you go to multi-QC, say multi-QC2. Okay, alternatively, you can add underscore to their respective names. That's also fine. So FastQC underscore 2, multi-QC underscore 2, flatten underscore 2, just to differentiate them. That's also fine. So the most important here, the takeaway message here is that we should have unique names, all right, for the various steps, okay, that we are using. Even if they are handling different data sets, we should have unique names for them, all right. So once we have this done, then we can proceed. Okay, that is what we have here, all right. So what we have to do is to come to save, and we can click on save workflow. So once we have it saved, now it's successful. So that means we can proceed with the exercise. And take note that to avoid duplicate names, you can also give it some description. For example, the flatten, FastQC, multi-QC, we can add raw, trimmed to also differentiate them, to let us know what data set they are using. But it's fine. Let's just keep them as it is. So now that you are done with this, you can click on play, which is this one. I call it play, but it's run workflow. So you can just run it, all right? So you can click on run. So when you click on run, you are going to see this coming up. Okay, you can see something like this coming. You can also, alternatively, another way you can also do it is that whenever you go to Galaxy, you can just come to workflow. All right, I'll just show that here. Let's say by mistake you close this. When you go to Galaxy, I'll just show you. When you go to Galaxy, the home page for Galaxy, you just have to come to workflow. When you come to workflow, you will see all your workflows. So this is the genome assembly workflow, and you can click on run workflow. So you can click this, and when you click it, you will still be sent to this. All right. So here you will notice that all the other steps or tools have not been shown because we have already linked them up. The only tool that we need is the one that accepts the inputs for us. Okay, so that's what we have with the FastQ. All right. So here, the FastQ that we are using dataset collection. Okay, so if everything's okay, it will automatically identify that for you. If it doesn't identify, just click and then select the dataset collection, the one you did. All right, and now you can run this workflow. So what you have to do is to click on run, and when you click on run, all those steps will run, and then you see. So you can also see the steps here, and as it's running, you'll be able to see which steps are running. All right, so here it tells us flatten, this one is running, one job successful is done, and then it tells us flatten, this one is also done. FastQC is currently being run. That is why we have the background this way. Okay, the background, I think it's orange or so. If it's orange, that means it's working, there's processing going on. If it's gray like this, that means it's in queue. All right, so green means it's done. So currently we see that some have 10 green, that means they are done. So we just have to wait for the others to complete. Okay, so what we need to do is to wait until everything has been done, and we're also going to see the outputs generated. Okay, so here we see outputs, lots of outputs here. Okay, And so here, what we can also do is that even this output that we have, we can also put them as collections and still organize them better. But that is for another tutorial. For now, we are going to see these outputs. So we have FASTQ, we have flattened, we have FASTP collections. Okay, so FASTP, these are the outputs. Okay, the reports, and then the output, that's the FASTQ files. This one is even done. So when you click this, you are going to see the FASTP outputs, which you can also download. These are the trimmed reads. All right, you can go back to history and you can check what else has been done. Now we have FASTQC being done also on the FASTP outputs. And after that, we have MultiQC, which is also being done. So all these processing are going to be done. So at this stage, you can even close your browser and come back and check it later. If there are things you want to do, you can go and do them and come and check later. So what you can do is to wait. I think mine took about 40 minutes for everything to get analyzed and generated. So you just have to wait for about 40 minutes or probably less. And then you are going to see everything turn green. Okay, so it's also running. I'll just go over again. So my analysis, the entire workflow, it took about 40 minutes to run. And so probably yours will also takes a similar amount of time or probably less. So all I have to do is to just wait until everything is done. All right, so this is what we are going to see. All right, so what you have to do is to wait and then just make sure everything is done. I think the one which takes the longest time is speed, because that's genome assembly. So genome assembly steps are usually they usually take long, depending on the data size and stuff. So you just have to wait for about 40 minutes and it should be fine. Okay, and then we can proceed. All right, the analysis has been completed. So you will see the output here. All right, so these are cool stuff. Okay, so congrats for successfully building a Galaxy workflow. So once you have this workflow, you can always reuse it. If let's say you have a different data set, you just have to make sure it's in the right format and you can just call the workflow and then use it. All right, so these are also the outputs. Remember, we ran FASTQC. So you have FASTQC report for the raw reads and the trimmed reads. We also run MultiQC, so you have the outputs also here. So all of them are here. All right, we also have FASTP. We run FASTP to have our trimmed reads. You can also see them here. Yeah, so we have this here. Okay, so these are the trimmed reads. When you click, you see them there. All right, so all these outputs are there. So now it's up to you to proceed with the downstream analysis or to make meaning of the data. All right, so what we did was a simple workflow for genome assembly. So if you are doing genome assembly, let's say for bacterial genomes, then one of the things you can also do is to evaluate the genome assemblies using tools like Quast, and then you can also check for genome completeness using tools like Bosco. You can also proceed further to do your genome annotation and then count the features, pangenome analysis, etc. So basically, depending on your objectives, then you proceed with the downstream analysis. All right. So for those who also want more information about the downstream analysis, you can always book a consultation with me, or you can book a session with me and then we can discuss further on that. All right. So that's for this tutorial. And so what I would like you to do is to watch these post genome assembly tutorials here.